Okay. All right. Yeah, who wants to give an update?
Hi, sir.
Hi.
Hello. Like, I don't have anything to present now. Like, I was mostly reading some papers and making my GitHub repository. I will share the repository today or tomorrow. There are some changes like I have to do in that. I will share it afterwards. The rest, I have nothing to present.
Okay, sounds good. Thank you. Looks like Shreyas wants to go.
Yeah, hi sir.
Hi.
I couldn't attend last bunch of meets, like last 2 meets I was unwell actually. Now I got time. I had some connectivity and all. Yeah, so recently I've been just working on just finding some new method just to just exploring from a curiosity point of view as to what can be done in this, like how can I approach this topic from a different point of view.
Yeah.
So yeah, this is not something like which is like I've implemented completely and I'm presenting. It's just something new ideas to look for this topic. So yeah, I can present.
Okay.
If anyone, so,
yeah.
Yeah. So let's start with this. Yeah, so this is just some unconventional and like new ways towards looking at the connectome development and its properties. So I found 12 ways, like 12 different things, but I shortlisted these three. Like one is like control theory, one is evolution game theory, one is like causal inference, and one is reinforcement learning. So like yeah. So like why these methods? This is just an attempt to explore different ways to look at connectome theory, just out of curiosity, nothing much. So I selected based on like these three criteria for usefulness, like does it explain the developmental law or help interpolate the connectome, or does it reveal a real property, or it's just like mathematical fitting. It should not be just a mathematical fitting because like models do sound fancy and all, but it should actually be some useful. The actual implementability, if like is the data required actually available? Can this actually happen? Can I run it or something? And then third thing is low abstraction. So like mathematical models and these like fancy theories tend to abstract things a lot. But I would try to have methods which abstract a bit less, because at the end I have to explain, like we need to explain the biological grounding and the actual biology, which may or may not come from the abstraction. So yeah, I would try to keep the abstraction low. So yeah, I have surveyed like 12 methods, selected 4. So data sets like this, a calcium data set, there's like the calcium imaging data set, the Whitwillet, and another data set I just didn't mention. It was of Drosophila. So yeah, this is just an overview. I'll come back to it later. Yeah, so first was the control theory of the connectome. So like there's a field in like electrical engineering which we learned in our college called control systems control theory. So like it was pretty amusing for me. So like how can we apply control theory here? So the question was the question which we were answering through this thing is like which neurons stimulated can drive the entire nervous system to any desired activity state. So like changes in which neuron cause the nervous system to drive towards some activity state. Since we are the like at the end interpolation of connectome is just a bunch of activity states. So like yeah, so these are called driver nodes in the nomenclature of this paper. Like basically I just reviewed a bunch of paper and I'm presenting it, and I also implemented some methods just as an exploratory thing. So yeah. So now why it makes sense for connectome development? Like this paper showed that biologically identified command interneurons of CLE, which are these ones, are exactly the minimum energy driver nodes predicted by control theory. So what control theory predicted is what are the driver nodes which, if driven through, will drive the connectome in a way it requires the minimum energy to drive. So what I mean by drive is just like shaping the connectome structure. That's what we mean. So like there is further mathematics which I have put in for the slides, but I'll just explain the intuition first, like why it makes sense. And so like yeah, so the development is like building towards a controllable architecture, not just any wiring, but that maximizes the brain's ability to coordinate its own activities. So I'll open up the paper as well. We'll go through that. So the developmental question here is, when do these neurons acquire controllability? When does it precede or follow synaptogenesis? So the figure here, I'll just come to the figure also. But yeah, so simplified math of it, it's a bit more complex. But yeah, to just explain from an intuition point of view, this was the maths of it. So like the connectome was modeled as a dynamical system. So there would be different ways to approach this. The way which I took in my proposal is the ODE way, the ordinary differential equation. Here it's also ordinary differential equation, but here they take it from control system's point of view, where they have a adjacency matrix for connectome, input matrix, control signal, control system to regulate my inputs. Now they have this controllability coming in, measures how easy it is to reach a state. So here, what I would try to do is, I would just try to cover the intuition of what each theory says, not going into exactly mathematics of it. Since I reviewed so many methods, I didn't go into exactly mathematics, but I just tried to see if some things methods are actually useful or not, and just explore things. So how you can apply this to it? So the minimum travel knows certain adult C elegans, or like exactly as I said, exactly interneurons. So what development can I do from using the thing which I'm doing? Like I can run controllability analysis on each of the 8 C elegans things, and like track if the average controllability for each neuron across time, like how controllable each neuron is. And we can just answer the question like do command, like we can answer questions about the structure of the connectome, as to exploring what each neuron does, just to infer, just to get an insight, deeper insight into how the connectome is structured. Like, do the command neurons have high controllability from birth, or does it build up? How does the build up happen? If there is a certain pattern in build up, then maybe we can use that pattern to interpret it and for like actually see if that pattern is followed or not. If the pattern is followed, we can like interpret according to that pattern. So like, is there a developmental stage where controllability sharply increases, which is the critical period in the development? Does controllability correlate with septogenesis timing for each neuron? So the questions like this can be answered through this method. In the paper for it, as you can see, so this is the model which they ran, and then they tried. So this was the control system which they got at the end. So these were its inputs, and these were the controls. This was the control system, the control nodes, and these were the actuators. So if we know from the basics of like mechanics and electrical engineering, that in each control system there is a control loop where there's some input, some actuator, and a control system like the plan transfer function and all. So similar things are followed in here. There are certain nodes where the controlling nodes, control system nodes. Certain were sensory inputs and certain were the actuators. So this is it about this method. Should I go into the paper, or like should I move on? Like, any questions on this method?
Not really. Just go into the paper and show us how they apply it.
Yeah.
This is network controllability. That's what they call it, right?
Yeah, network controllability. They call it that. So if you go to the abstract, just a second. Yeah, like should I go through the abstract, or like should I go through paper? Exactly.
Yeah.
Going through paper will take some time. So yeah. So if you see the important part, like so so what they try to do is they fill the gap by applying a control framework to the connectome of C elegans, allowing us to predict the involvement of each C elegant neuron in locomotor behaviors. So again, they down upon a certain part of C elegans, which is the motor neurons, the locomotor neurons, and they try to predict the involvement of each C elegant neuron and see how controllable it is. So we predict that control of the muscles or motor neurons requires 12 neuronal classes, which include neuronal groups previously implicated in locomotion by laser ablation. So as well as one previously uncharacterized neuron. So like, this is fine. So we validate that this prediction experimentally, finding that ablation of the certain neuron leads to significant loss of the dorsolateral polarity in the large body events. So I'll just go through the figure. The figures are quite informative actually. This is something which they ran for a certain set of neurons, and then they ran over the whole connectome, and this is the result which they got. They mapped out all the interneurons. The yellow ones are the controllable ones, which are the controlling ones, and this side would be the actuators, and the blue ones would be the input neurons. And yeah, going to this part, again, they just try to apply to various set of like here they show that how this neuron is uncontrollable and how certain neurons are controllable. The equation which they use is given in the starting of paper itself. So the dynamics of system composed of is described by this certain equation, and then there so this is a set of matrix and denotes the states m plus n nodes at time, and then these are the functions which capture the nonlinear dynamics of each node, and then these the v, this v factor, this represents the external stimuli applied to the test. So this is where input is taken through, and assuming that in the absence of the additional stimuli, the nervous system is a fixed point. And yeah, so more or less the larger part of the paper was trying to do this itself. Then they do some analysis, and like they in various visual forms they show the connection, like which neurons are controlled by which controlling neurons. So yeah, I would say this is most of the paper which they're trying to do. So yeah, I didn't go into the depths and exact mathematical intricacies of it. These are the parts which I went into. So yeah, this is for this method. Then yeah, I would move on to the next method. This is evolutionary game theory plus synaptic connection. So this started from a particular lab called Bhatt Lab and the Ganguly group from Stanford, where they are studying evolution and the theory of games. So yeah, so how do they apply? The setup is that two axons fighting for the same postsynaptic neuron. So each axon can invest metabolic resources to dominate the synapse. So this is explained from a Devan-Hock game. So it says so the investment is that if they both invest aggressively, both are weakened and neither wins stably. Okay, so before going into, I just say that for the setup, I guess it's not written here, but the setup is like there are two neurons and they are trying to get attached to two axons, and they are trying to get attached to the next neuron, the dendrite of the next neuron. So this attachment. So this is how the connectome grows. Right? The neurons get different neurons splitting and getting attached to different dendrites. So if a bunch of, so the way they are modeling it here, if a bunch of neurons are fighting to get attached to a dendrite, then how would the attachment happen? Like which one would attach, which one won't? So now if both invest, so they require some amount of metabolism and like some amount of energy to like move around and attach to dendrites. So if both invest aggressively, both are weakened and neither wins stably. If one invests, the other doesn't, and other doesn't, the investor wins and takes the synapse. This is the Hough game for the evolutionary biology, and it predicts synaptic competition outcomes. So why this is biology grounded? The competition is real. Like so the paper says that axons literally compete for the tropic factor. Okay, just ignore this short forms. And postsynaptic area. So basically, what that says is that the axons literally compete to attach to different dendrites, and the payoff matrix is real. The metabolic cost is measurable in terms of the synaptic territory is measurable, and how much metabolic cost is required, that is also measurable. So remarkably, the Hebbian plasticity, which is just, like I'll just explain what this means, falls out as the best strategy response. So this is a response for it, like how the attachment should happen. Not a separable assumption, but as Nash equilibrium strategy. So Nash equilibrium, again, is just a term in game theory, which specifies for a certain game. So this is a game like that of for this game. So this game, the Nash equilibrium comes from this state, this particular state. So Nash equilibrium basically weighs different strategies, and it assumes that each player is equally intelligent, and it comes up on a middle ground as to which strategy would be chosen by each player, such that their gains are maximized and losses are minimized. So that is what Nash equilibrium is in a layman term. So Hebbian strengthening, it says that the rational thing to do, if you are, so this Hebbian strengthening tells the rational thing to do if you are an axon and trying to win the synapse. So now coming to the maths of it, so the payoff function, so each game has a payoff function, and the Nash equilibrium is obtained through that payoff function. So V here, I will just explain the parameters of this payoff function. So the V is the value of synaptic territory, like how useful this postsynaptic neuron is. this is SI by summation SJ is a probability that exam I, this is the SI, wins the competition, and the metabolic cost of investment. So this term shows the metabolic cost of investment. So the Nash equilibrium here, the stable outcome is that if n goes to infinity, the overcrowded competition destroys all the values. And if there's n equal to 1, like there's optimal, like no effort, no competition. If n is 2, this is the standard case where one exam wins and one retracts. So in the evolutionary stable strategy, Nash equilibrium is stable against Newton strategies, and it's just which is biologically realistic. So now, again, not to get lost in all the terms. Like for me also, it was a bit confusing to get like lost in all the terms. We'll just come on the application part as to how we can apply this. Like, of course, how like something reasonably can be obtained through all this. So what this predicts in C elegans, right? So these are two interneurons, ABA, ABB. These compared for motor neurons and synaptic territory. So Nash equilibrium predicts the winner takes most pattern observed in the adult connectome. And the Angeli group says that applying this framework to C elegans, motor neurons predicts which motor neurons dominate without specifying a molecular identity. So through pure strategic logic, they are able to specify as to which neuron will go and connect to the motor neuron. So like which neuron out of AB and APB would go and connect to the motor neurons. And the MATLAB models through synaptogenesis as iterated game with incomplete information. So they model the synaptogenesis as an iterated game through incomplete information, and the replicator dynamics match the observed elimination curves in the mice. So I'm not quite familiar with this observation. I picked it out of their paper, but yeah. So basically, what the point I'm trying to make is through game theory and through just throwing these values at throwing these observations, which is there in the data sets. The data sets for this are obtained. By throwing these values, we can predict like which neurons will connect purely based on strategic point of view. Again, for this thing, the biological grounding of this method is a bit skeptical, not much clear. But yeah, like this is one of the interesting methods which I didn't expect to come up. So yeah, any questions on this thing? I know like this method is a bit bizarre for me. It was quite surprising, but yeah, I just found it interesting and I thought I should mention it in my presentation. So yeah, any questions on this part?
Yeah, where are these papers? Where are you getting them from?
Yeah, this thing. The references are here.
Okay, here we go.
Yes, I'll show this thing. I'll open it up.
So yeah, that was yeah.
Just a second. Right, so this is the paper for this. Let's go through the summary once for this. So yeah, so this is basically, the name is like one-dimensional dynamics of attention and decision making in LIP. This is where we allocate our visual spatial attention depends on a continual competition between internal. So this paper does it from a point of view of the perception, the part like the visual spatial attention and perception of that part of the connector. So yeah, so where we allocate our visual spatial attention depends on a continual competition between internal generated goals and external distractions. So recently it was shown that a single neuron in the macaque internal interpartial area LIP can predict the amount of time a distractor can shift the locus of spatial attention away from a goal. So they propose that this remarkable dynamical correspondence between a single neuron and attention can be explained by a network model in which generically high dimensional firing rate vectors rapidly decay to a single mode. Okay, so can I just come back on this after a while? I'll just once look it up once again, like by the resources, because I made the slides like 2 weeks back.
Right.
And then I intended to present it like 2 meetings back, but then it got delayed. I'll just go back. I guess I took some things from the book as well, the thing which is like evolution and game theory. So I'll just look it up once and get back on this. I'm not really clear on the like exactly the things like So this thing is the Bhatt et al, which I was putting the image, wanting to put here. So that also, I guess I took from their websites and from their articles, but I'm not really, I'm not able to find really right now whether it's so I'll just get back to you on this thing in a while. I'll just continue the presentation for now. So the third method which I was just exploring is the causal inference and the calculus. So what is the problem here? So what they're trying to do is the C. elegans is the largest laser ablation literature in biology. So ablation is like thousands of experiments where killing a specific neuron and measuring the behavior. But every paper reports correlation, like when ABA is absent, backward locomotion fails, something like this. But correlation doesn't imply causation necessarily. So what this means is basically this thing. So ABA might be absent because another neuron are degraded first. So the causation direction is unclear for this. So what does Pearl's do calculus does is like, is it the right language for ablation experiments? So what the biology does is like probability of the certain behavior, observing a certain behavior given ABA is absent. So the passive observation is that ABA could be absent for any reason. Now, what the do calculus does is the probability of behavior given that we kill the ABA. So the intervention is we force ABA to be absent and all other variables are free. So this is what laser ablation experiments actually is. So what the laser ablation does, it kills the neuron, and the observation which the biology does is like ABA is absent. So ABA being absent and we forcibly killing ABA aren't actually the same thing. Like they are related in some manner. They can be same. Like this is like a subset and like a superset and a subset, something like this. So ABA can be absent. Its reason may be that ABA is killed intentionally, or it's just not present and some other neuron is taking its part, like some other neuron degraded first. So yeah, so this is the intuition of it. This is the framework for it. So first I'll go on to this part, like how can we apply to it. So this is a paper which has the whole brain's calcium imaging data in C. elegans. So 107 neurons tracked simultaneously during spontaneous plus local activity. So this is something from which we get the activity of brain from the calcium imaging data, locomotor activity. And then through this paper, so this freely behaving, this has the imaging of freely behaving whole brain imaging. So this is a neural activity plus behavior at single neuron resolution. So we have quite a lot laser ablation data, which is like interventional data which already exists. So we can run PCMCI, which is like this method, this algorithm, which is the causal discovery in time series. To get the causal graph, G causal. So to explain what a causal graph is, I would like to change to this thing. This is actually a report which I was going to present. This is a report which I made, but then this was a bit too long, so I just shifted to like this had all the 12 methods, but then just I decided to shift to the PPT presentation. So again, to explain the causal thing in a better way. So yeah, this is the thing, is the intuition which I was going through. This is the mathematical parts of it. Now coming to the application of connectome. So the developmental part is the developmental translation. So as C. elegans connectome matches through 8 stages of the dataset which I have. The causal graph structure changes. So the early stages have weak causal edges, and the mature stages have strong directed causality. So this is the main part of it. So this is what we can get through the method as to what part has, like initially the stages are weak in a causal manner, and later the stages become mature and the causality is quite evident. So applying this method to the calcium imaging data set and these calcium imaging data set. So this one is of neural activity and other is of the motor activity at different developmental stages, but produce a time series causal graphs. So comparing these to the structural connectomes gives an answer to the fundamental question, like when in development does wiring becomes causally functional. So yeah, that is something which this method tries to do. But yeah, so again, coming to the okay, any questions on this method?
No, I get it. Yeah, it's using causal modeling in the network. Yeah.
right. So basically, we are trying to differentiate between these two things, like how does behavior change when I change something, and this gives me a causal graph across different time steps, and it helps me understand across different time steps. So yeah, so these are the resources for this. So yeah, again, I'll come to the last part, which is the reinforcement learning and synaptic competition. This is closely related to the game theory part, which I was telling. So yeah, coming to the basic introduction of RL, like what RL would do. So there's a in every RL problem there is credit assignment problem. Like an agent takes actions over time and gets rewarded, and it asks like which past action caused the good outcome, and the credit is assigned, right? So in connectome, what we can do is like multiple axons make transmission attempts, which is like signal transmission. Some wiring configuration produce functional circuits. Now, if a circuit is functional, it's a reward, and if it's not, it's not. So now, basically, I have a connectome at time step t. Now, I can that connectome can go to any state. For the like, I have state t1, t2, t3. It can go to any state. I'm at t0, right? But now, which state is actually useful? So if a certain state is functional, then that state is good. So like a certain wiring, a certain changes in my wiring, a certain connections made my state useful. So that is a positive reward. And if it doesn't make it useful, that's a negative reward. Right. So this is the way through reinforcement learning which can be applied. So now, according to this paper, like inside, the Hebbian plasticity is reinforcement learning. So what they say, so here again, a bunch of buzzwords and like terms are being thrown, stochastic Hebbian learning and all. But again, to not get lost in the terms, what I'm just trying to make an intuition and this exploratory part, as I told, like again, nothing much like the modeling and like mathematical here. So again, to tell is that the neuron that fire together are like when they wire together is mathematically identical to reinforcement policy gradient algorithm. So each synapse is independently running a policy gradient update. So its policy is whether to transmit. Its reward is downstream circuit functional. So each neuron in my connectome will have a policy of its own, and it will try to maximize its own reward. Again, this is similar to game-theoretical approach where each neuron was discretized. What we study, what I was studying in my proposal is the whole ODE as the whole system working together. But these two approaches, the reinforcement learning and the game-theoretical approach, is trying to model each neuron in its own, where each neuron tries to maximize its own reward. So, yeah, first coming to this part, the reinforcement learning, the application part, is that what this explains biologically is that ocular dominance. I just go to this part for the reinforcement learning part. I will go to this part. Yeah. So this particular paper evolved the C. elegans like locomotion controller in simulation using a genetic algorithm, which is a type of like populated level reinforcement learning. So finding that the evolved circuits replicate the motifs from the real connectome. Motifs is something which I've used previously as well. These are the small patterns which we see. So the gap is individual. So yeah, one of the useful finding is in this paper the C. elegans like connectome controller, C. elegans a locomotion controller in simulation is done using the genetic algorithm. This genetic algorithm, the population level RL, and finding that the evolved circuits recapitulate the specific motifs from the real connectome. So they tried to build the connectome using an RL method, and they tried to verify using the motifs which actually made sense, and the real motifs were present in the connectome which they got. So the gap is individual developmental RL. So running a structural RL agent whose environment is C. elegans body simulation. So this is a very ambitious thing. I get it, but yeah. So this is something which can be done. So yeah, this was the overall presentation. Yeah. Any questions on the RL part?
So you're basic in Izquierdo and Beer. If you go back to that paper, that's where they're modeling C. elegans movement circuits. And so what they're basically doing is they're implementing a population level reinforcement learning, where you basically have a population of neural or movement circuits, and then you basically have some individual account of that. So like you're selecting the best motor programs, basically. Is that correct?
Yeah, I would say. But, yeah, I would say that does make sense. It's similar to what they're trying to do. So yeah, I would agree with that. Um, and yeah, this is something which I tried to like unify the four methods, but I don't think it makes much of a sense. But yeah, like game theory somehow tells like which synapse would compete and like which would survive the competition. Reinforcement will tell like how each synapse is getting selected. Control theory would tell like how to be like what are the outcomes of my achievement, and causal theory like does the result actually work? Like if I remove a neuron, how does the things change?
Yeah.
But yeah, this is the overall structure which I was thinking. Like again, this is not something like what I proposed was way more like I went into mathematics of it, but this is just a exploratory thing. Just a honorable mention to put one method which I found quite amusing in all of this. So I did put it here, but then I ended up not putting it in my um this thing. Yeah, this was one of the method. So what it basically did is that this was an information geometry, like information theory, something which came up from the information theory. So they said that how to say that how different are two connectomes. It would see how statistically distinguishable are the probability distribution that generated this connectome. So this is the main part basically. So this was modeling the probability distribution of a connectome being generated, and then it will differentiate between how different are two connectomes and like how so this differentiation was based on the probability distribution of each connectome being generated. So they used like many methods, many mathematical techniques like Markov chains and all in this to get this probability distribution. But again, I didn't find this much of a use because what would we do from finding like how much different are two connectomes? But yeah, this was something which was quite interesting. But I didn't include it in the final thing. So yeah, this is it for today. But yeah, I agree that this was not much well thought, like well mathematically formed. But yeah, I was just exploring through stuff.
Well, that's
so yeah,
good.
thank you for listening.
That's good. Actually, go back to your slides.
Yeah.
Yeah, I want to see that. So this slide, could you do a slideshow? There we go. Okay, so that's game theory, reinforcement learning, control theory. So you're basically trying to characterize the individuals. Well, you're trying to characterize the interactions, then maybe the individual strategies, then the sort of the way in which it's controlled, and then causally controlled. So, I mean, yeah, this is a nice sort of chain of things that you would use to explain different aspects. It almost seems like reinforcement learning might come before the game theory, because you have reinforcement learning for the individuals. They set up their strategies, and then the game theories where the strategies are implemented. Then the control theory might be like where the strategies are, how do they get controlled, like at the level of the strategy. So your reinforcement learning is where the strategies are being selected as viable. Game theory is where it's being implemented in an interaction. Then your control theory is kind of explaining that, and then causal inference is saying what is causal and what is maybe correlative, because we often don't know what is causal. We have to do these experiments where you have to maybe ablate something or do an intervention where you have like some action on the system as an external observer. So, this is a nice, if we could kind of figure out how this fits together, and if you had, like, say, for example, like an individual or maybe a population of individuals. Let's take the example from the genetic algorithm. You had a population of individuals, and they all kind of course, there's variation in how they achieve something, and that's kind of the point of evolution, is that you have variation, and then it gets selected or it gets bottlenecked through some sort of drift process or something like that. But basically, you have this variation in the individual agents. That's like going to be, you could use reinforcement learning to characterize it. You could use a genetic algorithm to characterize it. You could use other kinds of methods too. but that, you could swap that out. And then you have the game theory where you have your strategy suite, and the strategy suite would be maybe continually updated from the reinforcement learning. So we don't know, like in biology, we don't necessarily know all the possible states, all the possible strategies. We might generate new ones. And then the game theory would test that out and see which ones are stable. And then control theory would be building on top of that, where you'd have a controller. Like in the controllability literature, they're really looking at what neurons, in the paper that you showed, what neurons or what nodes control other nodes. Basically, which nodes are the hubs in the network, and then those control that set of interactions. So you have this kind of interesting connection between control theory and game theory, where you could use controllability, or you could use some control, strategy. Basically, you're looking at how game theory is working downstream. What nodes are being affected by a certain strategy being implemented. There's also this connection between control theory and reinforcement learning too, because reinforcement learning is where you are kind of reinforcing different strategies, or you're reinforcing a policy, and you're saying, if this policy is pursued, it's rewarded, and then that's kind of the one that's selected. And so in control theory, you're also kind of making sure that feedback is reinforcing the proper, the system being within the proper parameters. So there's that aspect of it too. And then causal inference is like narrowing down everything and saying, what are the causal relationships there? What is actually causing what, or is it just kind of like, random? You could have like a random reinforcement learning and a random control theory. It wouldn't be very good, but in biology, it might generate something interesting, and we can't say whether or not something is causal. So this is nice. This is a nice architecture for this. And then there's all this math that you have to do to kind of, but I think actually
that is the math part is the difficult part. So like in my proposal, I just explored the hyperbolic space, which is like one math part, and I explored that in depth. These are like 4 things. So like if I have time like later, and like if I get selected, and like later if I have time, I'll have to like explore all of this and like connect everything in depth. So definitely, I would be interested to do this. This was just an intuitive and a overview of everything as to what something can be done from a different point of view. Like while making my proposal, also I felt that okay, I'm going deep in one thing, but what if there's something else which can model same thing? Like of course, biology is something which came about itself. Like this is not which is model. Mathematics is something which we develop. So we are trying to fit biology in different models. So that is what was coming to our mind. I'm fitting it in one model, but what if some other model fits it better? So so this was just a try to find other models where I can fit biology in. So if I have time later, I'll of course try to explore all these models and just try to get something good out of this.
Yeah, I almost think it's more interesting to look at, like, instead of looking at the math, look at like some sort of simulated biological system, like create a agent-based, almost like an agent-based model or something with agents in it, where you're just doing that. And you don't necessarily have equations. You can maybe work out the equations, but like just demonstrating how it works in that sort of model, where you have a population, you have variation, and you have these things going on that kind of control the, or how these things play a role. Because, people will, sometimes will do papers where they make claims and say that like biology is doing reinforcement learning, or biology is doing, reinforcement learning comes from biology originally, but so that's kind of obvious, but like it's doing game theory or it's doing control theory. And of course, the idea is that's a theory. It's not really a thing that we know is biology. We're interpreting that from the biology. So, but we could build agents that like say, okay, this is how an agent would behave under this theory, and it fits pretty well. And I think it's a lot more effective than, just kind of saying this is supposed to work. Yeah.
Makes sense.
All right. So yeah, this is great. Thank you.
Yeah, thank you for listening.
Yeah.
And yeah, sorry for the like I did miss on some details. I didn't go into like some details of some stuff, but yeah,
It's okay. That's okay.
Okay, thank you.
You're welcome. Yeah. So yeah, I have actually here. I'll show you something here. All right. So I have this working paper. This was from 2022. I actually presented this at Dynamics Days, which is a dynamical systems conference, and it's like just a working paper. It's basically this game theory of developmental processes. It's a long review of like game theory in development, and it's really about not necessarily connectomes but developmental biology. So it kind of goes through some of the things that we have to think about in game theory and in games, and there are certain things about development that are important here. um, and we go through. I think the way I structured this was that it goes through 0-player games, n-player games, and games that generate larger systems of interactions, equilibrium states, and computational complexity. So kind of good, and it's very simple. It doesn't really have a lot of. It didn't do any kind of agent-based modeling or anything for this. So it was just kind of a counterpart to that talk. But I'd like to actually, we should write a paper on like kind of this larger space of game theory. Where we're just kind of thinking about how would we implement games in development? Like what is like you were talking about how we have an opportunity to sort of interpret some of these interactions as games, with synaptic formation and other things. But there are I think a larger set of questions or issues that you could use game theory for. Like there are a lot of interactions in embryogenesis where that are basically could be modeled as games of some type or another. Yeah, I mean that's great stuff though.
Yeah, sure. The paper on this was very interesting.
Yeah. All right. Yeah. So thank you, Shreyas, for that. Did anyone else want to talk about anything they have going on? Or
Hello.
hi.
Yeah, okay. Yeah. So I went through some papers, and except that, I didn't prepare a presentation as such. But other than that, I would also like to give an update. Like apart from neuroscience, I also focus on AGI, and I look into those things as well, like on the proceedings of those. So I was helping an organization called Blue Dot Impact, which deals with education, giving education on biosecurity, AI safety, and alignment to different organizations, and letting them know the risks of AI. So I was helping them shift up, like make a few courses and stuff, maybe with the documentation. And so that's what I was doing for the past week. And as a token of appreciation, they're also sending me a book.
Oh, yeah.
Yeah, I like reading books, so I accepted that.
Good.
So.
Good. That's great.
Yeah, the book is called The Alignment Problem. It was a New York bestseller in 2021, and it deals with how AI can learn human values in the future.
Yeah.
So yeah.
Great. Thanks. Yeah. Sounds great. Did we have anyone else? Ashu, did you want to say something? Or okay. Susan?
No, like I said, I'm trying to sort myself out here. I've collected a bunch of papers, and some of them are relevant, so I'll try to collect 100 of those, and the rest can be burnt or something. Yeah, and I apologize. The screen here is blurred because the background's blurred. I don't know.
That's fine.
Anyways, I have a blurred screen. Anyways.
Great.
Yeah, I'm looking at right now at a paper called Spatial Segregation Between Cell to Cell and Cell to Matrix Adhesions.
Oh, wow.
So they're at a point, they're not spread along the surface, like they're at specific points. So that goes in with the modeling that I'm doing.
Yeah.
They're just attached at specific points. And I've heard that hydrolonic acid was in between the attachment points, and I'm not sure about that. But that's what I heard in a lecture one day at a physics conference.
Yeah, all the hydrolonic acid.
Yeah, I don't know. Yeah, so the focal adhesions, especially between the cell and the ECM, the basement membrane, they're not attached. They're just attached at the adhesion points, and that's it. They're not in between. And at that scale, you've got they will attach to each other unless there's something like hydrolonic acid in between.
Yeah.
So that they can't attach.
Huh.
Because normally you have van der Waals forces and so on, and the Casimir, even the Casimir effect, where you've got the two surfaces, they tend to glue together.
Right.
Yeah. So I don't know what's going on with that. Anyway, that's my question of the week, I guess.
Yeah. That's great. Yeah, thanks. Well, we'll have to present some week on that. So.
Okay.
Great.
You want me to give you this reference?
Yeah, just type it in the chat.
Type it in the chat. Okay.
Yeah.
Oops. And I've got paper copies as well. I was having trouble reading from my computer screen, so I wasted a bunch of money printing some of them out.
Well, Susan types that in. Why don't we get on to the final thing for today? So I had a couple papers I wanted to talk about, just briefly. We'll kind of go over this, and they're related. Like last week, I want to relate them to sort of some of the things we've been talking about in the group. So this is from ChemSystemsChem. I'm not familiar with this journal, but it's a chemistry journal. It's from Chemistry Europe. This is called Higher Order Behaviors in Bio-Inspired Materials. So this is the graphical abstract here, where we have these higher order behaviors in chemical systems. So we have these materials that we're building. We have these different types of molecular systems. And the idea is that we have these higher order structures or higher order behaviors that we can look at. And like this is just a review of a lot of these different classes of things that people might be interested in. So these higher order behaviors are four kind of categories. There are the polymer networks is the first category. And these are networks of polymers, like you might have like actin or something like that, or some protein structures. And they're connected together as a network. So they have nodes and they have edges. The edges are usually the polymer stretch, the polymer length of the polymer, and then there's some attachment to each other, so they form networks. So like actin networks would be a polymer network. And then you have these structures, but then you also have the behaviors associated with them. And so that's I think is really interesting, and what they're trying to tell people about here in this review is that you can build these structures, but you have to build them to optimize for certain interesting behavioral properties. So in polymer networks, we're interested in self-assembly, which is where they can self-assemble or self-assemble into different configurations. So like actin networks, we know, are reconfigured in when cells are remodeled during migration, during formation of a sheet, during times when the cells are under stress and strain, and all of that. And so understanding that self-assembly process is important. It's also important in morphogenesis because there's an assembly of these kind of networks during morphogenesis, during, say, like cell division. Then there's self-healing ability. So in a lot of material science, we're interested in self-healing. So how do materials heal themselves? If there's especially like things that are biological in nature, like if we're building like cell sheets and we're engineering cellular sheets for various reasons, how do we control self-healing? So we know that biological systems can heal. We know that like if we, sometimes a cell can die, but also it can heal, or a cell sheet can heal itself. It can divide and fill in the gap and heal. Or sometimes a actin network can reconfigure itself to become to recover its function if it's disrupted. And so, how do we build self-healing materials? This is very important for like things like implants. If you're implanting cells into someone to fix things that are wrong with them, or if you have biomaterials that you might use for other purposes, where they're under stresses and strains, and sometimes they break, and you don't want to have to replace them, so you just allow them to heal themselves. And then contract.
May I add something?
Yeah.
If the material that you add, doesn't matter whether it's a cell or artificial material, if it isn't the same, has the same elasticity and other mechanical properties as the tissue surrounding it, it will form a scar.
Yeah.
Because it doesn't move with the material. So it's a scar, becomes scar forming.
Yeah. So yeah, that's true. And we would like to not do that, I guess, in a lot of biomedical applications. And then there's contractility. So again, being able to control the sort of material properties of the contractility of the material. And this is, goes back to this network controllability thing we were talking about earlier, where if you can understand the constituent nodes of the network and how the topology controls like the entire network, if you have, say, hubs that control sort of how the network is, moves or shapes or allows for behavior downstream, then we can have a better understanding of how these other processes work. So those are polymer networks. They're microbots then, which are these little machines or these little agents, autonomous agents that are at this molecular scale. And we might have, say, like mechanical microbots. We might have microbots that are cellular in nature. We might reprogram cells to be these autonomous things that do certain tasks in a biological context or in a chemical context. And there are all sorts of things, behaviors that microbots can perform, and they do this not just individually but collectively. So you have these macroscopic taxis. Taxis are movements, very simple movements that small agents will make towards a source in the environment. So chemotaxis is where agents will move towards a chemical source. Phototaxis is where they'll move towards a light source. And so there are a lot of different taxis that we can, our microbots can interact with, and we can program them to interact with those in a certain way. But they also exhibit these collective behaviors like swarming and collective action, where, of course, they will do this if you give the agents the same instructions in parallel. They'll tend to form these collective structures, and the idea is that we want to be able to kind of control swarming and other forms of collective action, which is hard to do, but if we kind of know what the input behaviors are, we can kind of have some handle on that. So these are things, again, like our program systems that are going to exhibit these kind of emergent properties, and so we want to be able to get a handle on how to control that. And it's very different from controlling the individuals in parallel. Then we have proto-tissues, which are these enhanced sort of sheets of maybe of cells or other types of things. Proto-tissues, they kind of look like two cell sheets that are like stuck together. And these two cell sheets in this example can bend, so they can go from like forming a linear array to forming a curved array. And the idea is that this is sort of the beginnings of a tissue. So if you're doing tissue engineering, you'll want to have sort of the component parts of a tissue. You want to figure out, what do I need to put together to get a tissue? So like we've talked about organoids, we've talked about epithelial cell sheets. Now, all these things that we're talking about with respect to adhesion and respect to cellular physics and tensegrity, that's all sort of part of this proto-tissue process where we're trying to figure out how a tissue behaves. How do you structure a tissue to be like an actual biological tissue? And of course, this is important in bioengineering, where you need to figure out how to incorporate like multiple cells or groups of cells into a matrix and get it to behave like that tissue. It's also important, say, if you're developing tissues de novo, you're kind of, people have engineered things like livers and hearts. They're trying to do this, and they're trying to figure out the ingredients. So there are a lot of things, though, in proto-tissues you need to understand. You need to understand what the enhanced properties of these proto-tissues are. That is, what are the properties of cells in sort of this array when they're in this collective array versus when they're just individually interacting with one another. What are the macroscopic changes? So what are the changes that occur when you have a bunch of things arrayed in sort of a sheet? There are these macroscopic properties and then changes as you bend it. And then, of course, there's this long-range communication, so communicating from one end to the other of this sheet, or communicating from this sheet to this sheet. So from the orange sheet to the blue sheet. That's important to understand, because then you can understand sort of the nearest neighbor communication, but long-range communication is more mysterious, I guess, and it's important to know because it's often nonlinear. Then finally, we have the protocell model. This is something that people use not only in biological engineering, but in origin of life, where they're trying to figure out how the first cells formed and what components you can boot up a protocell with to get like a minimal cell, which is basically the smallest number of components that form sort of a functional cell. And so what people do in protocells is they'll have these vesicles, which are just these empty kind of bubbles with some with a membrane, and then they'll boot it up with things with sometimes with biomolecules. Sometimes they'll try to put a genome in there, and so eventually you can get like a handle on what these minimal cells can do, what they look like, and then map that to what cells, maybe like a eukaryotic cell or a prokaryotic cell is actually doing. So there are a lot of things in protocells that are these higher order behaviors, including cell division, cell assembly, macroscopic taxis, like we saw up in the microbots example, and ecosystem-like dynamics. So protocells kind of living like in these proto-tissues and how they kind of start to learn how to cooperate and proliferate and so forth. So this is I'm not going to get into this paper, but I just wanted to show you sort of this graphical abstract. I think it summarizes the review quite nicely. So it's a nice, it captures the state of the art of this area quite nicely. And so, if we were thinking about something like game theory or something like that, we could easily use this as like kind of a basis for game theory. So a lot of these polymer networks or these ecosystem dynamics and protocells, they can be characterized by game theory. The idea is that the cells have like this imperative, maybe to reproduce or to connect to one another, or to maybe there are cells that or nodes in this polymer network that become sort of the controlling nodes through interactions, through their location in the topology, through some property in that cell that makes them kind of the controlling cell. And we don't know what that is, so maybe game theory can suss all that out for us. Then there are other mathematical models we can use to model this as well. This is a second paper. This is kind of moving in this direction of smart materials and material science plus, you might call it. And it's where we're thinking about the sort of the macro effects of materials and how we're engineering more biological materials that have the sort of where you go from like the constituent parts of materials, where you go from that sort of reductionist view to this sort of emergentist view of cells behaving collectively and these biological systems, these autonomous biological systems. So this paper is called Material-Based Intelligence, and I think this is just it's from the archive. So this is from the Condensed Soft Matter archive, and so this is from 2025, so it's not that old. This is called Material-Based Intelligence, Self-Organizing, Autonomous, and Adaptive Cognition Embodied in Physical Substrates. So this is the idea that there's this sort of maybe autonomous cognitive aspect to materials. And cognition is used as a metaphor here. But there's also the self-organizing and autonomous aspect to it. And so this is where they kind of go over this sort of, I guess it's a hypothesis paper where they're kind of thinking about how we can go from these simple pre-programmed stimulus response behaviors to these autonomous behaviors that are sort of goal directed in nature, I guess, or cognitive as they think about it. We did a paper on this in this area in 2023. We have our own views on it, so you can go look at that paper. But in this paper, I'll go through the abstract and then we'll kind of ruminate on it for a bit. So the abstract reads, the design of intelligent materials often draws parallels with the complex adaptive behaviors of biological organisms, where robust functionality stems from sophisticated hierarchical organization and emergent long-distance coordination among a myriad of local components. Current synthetic materials, despite integrating advanced sensors and actuators, predominantly demonstrate only simple pre-programmed stimulus response functionalities. This is falling short of robustly autonomous intelligent behavior. These systems typically execute tasks determined by rigid design or external control, fundamentally lacking the intricate internal feedback loops, dynamic adaptation, self-generated learning, and genuine self-determination characteristic of biological agents. So this is interesting. They talk about this, there's, of course, external control, say from an experimenter or from like another system, and you have these design parameters that are very much like what you might find in like a refrigerator or automobile. And then what they're saying is that we want to create materials that are actually autonomous, that have their own internal states, that have their own sort of adaptive states, and then the ability for these materials to sort of behave like biological agents. In other words, you leave them alone and they can do their thing, and you don't need to have much external control at all. Of course, there are a lot of things that need to be in place for that. There are a lot of implications of that. There's a paper by Levin and Bongard, which we've talked about in another group, called Living Machines, and they grapple with this idea that a lot of the machines that we're building are not just sort of mechanical devices, that they're actually living systems, and they were with a lot of the biological engineering efforts. We are actually breaching that sort of boundary and saying that we have these living systems that we're struggling to sort of control. through our typical engineering lens. And, it's not as simple as just applying engineering methods or mathematical models. It's that you have to understand what these systems do and then kind of learn from that and build, sort of characterize them accordingly. So this is what I meant by, like, you don't necessarily want to use equations. You might want to build a agent-based model first to see what exactly, what the sort of the way in which these things behave, and then kind of work backwards from there to kind of characterize this mathematically. But in any case, so yeah, and then they say this perspective proposes a fundamentally different approach, focusing on architectures where material-based intelligence is not pre-designed, but arises spontaneously from self-organization, harnessing far from equilibrium dynamics. So this is where we have this idea of almost like chaos theory, or chaotic systems, or chaotic control. So it's like there's control theory, which of course you have first order, second order, third order control theory, and then you have this sort of far from equilibrium behavior, which of course you have to use then a different class of control methods that involve chaos. And it's a different ball of wax. And of course, if you don't understand that you're dealing with far from equilibrium dynamics, you tend not to use those methods. But that's again something that it's kind of typical of biology. It's typical of maybe some of these materials that we desire, but of course, we can't just imbue a material with far from equilibrium, or a material substance with far from equilibrium dynamics. We just want to be able to design a system that's sort of has autonomous properties, has these collective behaviors, and then that's how we would characterize it, using these far from equilibrium models. It's basically where there aren't these discrete states that we can count on, that the behavior moves around the state space quite a bit, and it's hard to say that something is in equilibrium or that something is sort of in this set of states that we can characterize with a Markov model.
That's why I think the tensegrities I was working with are far from equilibrium.
Yeah, that's exactly
Yeah.
right, and that's
It kind of matched the biology in a way.
Yeah. So such approach leverages minimal physical models, intrinsically embedding information theoretic control in the materials and physics, and its seamless coupling with the environment. So again, we have minimal physical models. We have information theoretic control. So we have kind of control theory, but it's information theoretic in nature. And then we have this coupling with the environment that they say seamless, but it really is about looking at the interactions of the environment, that basically it's not going to that it adapts to its environment when needed. This work explores interdisciplinary concepts from material physics, chemistry, biology, and computation, which we talked about, identifying concrete pathways towards developing materials that not only react but actively perceive, adapt, learn, self-correct, and potentially self-construct, which is a self-assembly process, moving beyond biomimicry to cultivate fully synthetic, self-evolving systems without external control. This framework outlines the fundamental requirements for and constraints upon future architectures where complex goal-directed functionalities emerge synergistically from integrated local processes, distinguishing material-based intelligence from traditional hardware-software divisions. Again, this is where you have, if you're a lot of times in materials science or in bioengineering, we've used these hardware-software analogies where we have the hardware, which is the biology, we have the software, which might be like engineering a genome, or controlling things with chemical, like with things like cell culture medium, or even substrates, different types of substrates. And then that would be the kind of division. We couldn't necessarily, there would be some interaction, but we wouldn't understand how to control that or even what was going on. We just had to hope that, like, say, we had the right substrate or we had the right medium or we had the right, we hit the right gene to modify. But of course, this is all kind of, without having access to this sort of this interactive system, this feedback loop, and being able to control things more precisely that way. This demands that concepts of high-level goals and robust, replicable memory mechanisms are encoded and enacted through the material's inherent dynamics, inherently blurring the distinction between system output and process. So, materials have memory. They have this sort of aspect where they can retain their shape under stress or whatever. They can maintain other properties through perturbations. But, I guess the idea is you want to build in a more robust type of memory. So going from like something that is, kind of material shape memory to something that is more cognitive. And so this is where, you're dealing with Hebbian learning and things like that. So this is another kind of vision. It's sort of a hypothesis idea. And, it's a hypothesis paper. And so, again, it brings up a lot of interesting points, a lot of interesting questions. And so this is kind of the state of this area. It does relate to biology, of course, but it does also kind of ask us to think about these other methods, like what Barshan or what Shreyas was talking about, and be able to kind of grapple with this. Any comments or questions? So this is the paper that Susan put in the chat. Spatial segregation between cell-cell and cell matrix adhesions. This is current opinion in cell biology. So that's from 2012.
I spelled segregation wrong.
That's fine. That's fine. You'll find the paper. But that sounds a little bit like agent-based modeling, actually. I mean, that's kind of what the classic kind of result that they get in agent-based modeling is you can get like spatial segregation within a model or a random state, and you just have to run the model based on its interactions and its current states, and it'll just update, and eventually it will separate out into like 2 stripes or 3 stripes or something like that. It's kind of like in morphogenesis with something like the French flag model, except the French flag model is just kind of assumes that you have these local interactions that are kind of guiding this gradient, and then there are these boundaries between the gradients, and you end up reinforcing that. I'm not quite sure that's how it works in the agent-based modeling example. I think there's no assumption of like a gradient or of any kind of control like that. It's just kind of like the striping or the the sort of regional homogeneity results from interactions, just kind of being locally reinforced.
Yeah, I was listening to video about Hydra and how it creates its head and tail depending on the actin alignment.
Yeah.
Dick told me to watch it. And then I was also looking at bacteria, and if you have a fast and a slow-moving bacteria, you get the fast-moving bacteria are slower because they tend to move together and then end up upright like this and then they can't travel. And apparently you get all kinds of interesting structures that can form around these clumps of bacteria or clumps of tissue or cell cytoskeleton or whatever you're using.
Yeah.
So anyway, I just took hand notes from that. I should send that to you because it was kind of shows what happens at that type of microscale, which is different from nanoscale and different from macro scale. Like it's its own environment.
Yeah. Oh, you remember for a while we were talking about condensates in cells, and they kind of do the same thing, where you get these basically, like proteins or other types of biomolecules. They aggregate in the cytoplasm. They form these aggregates, and then the aggregates do things for the function of the cell. Sometimes they aid, like ribosomes. Sometimes they kind of serve as aggregates for, like, I don't know, storage sites or things like that. They just have a lot of different functions. People are kind of exploring that, like, what do they do in the cell? And, it might just be this organizational thing where it's doing things locally in the cytoplasm. And, if there maybe were like a second great transition in life, we'd have like super eukaryotic cells where the aggregates, these condensates, would serve as like maybe another class of organelle or something. I don't know.
But. Well, they're calling them plus half defect and minus 1/2 defect.
Oh, in that paper? Okay. I can't really see that.
I can see that.
Yeah, it's blurry, but that's okay.
Anyway, it's on YouTube, and it was the first one is a SLAM meeting, so.
Oh, okay. It was from SLAM.
Yeah, SLAM, and then a related one. So about rosette formations, for instance.
Yeah.
I'm interested in rosette formations.
Oh, yeah. All right. Thanks for attending. And I look forward to doing some additional work on this, or, seeing what we can do. And, all right. See you next week. Thank you.
Hi, Bradley. One thing I wanted to ask. Can you please tell the name of the paper which is showed for the game theory? I would really like to go and read it up, like the one which was authored by you, which is screen shared.
Oh,
Like,
yeah,
what
I'll
was
put that in the chat. I'll put that in the Slack, so we can do that.
Sure.
All right, yeah, thanks.
Yes.
All right.
Thank you.
Thank you. Bye.
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 Bye, sir.
Bye.